Welcome back, everybody, to the second part of the respiratory system lecture. We have completed in the first part of this lecture material the gross anatomical, tissue, and microscopic anatomical characteristics of the respiratory system. We created all elements of the conducting system, the respiratory zone. So we have an anatomically perfect respiratory system ready to go. At the end of the first lecture, we looked at the need for moving air through the respiratory system. This process, ventilation, is the next big topic. And then we will move into more molecular terms, just to explain how oxygen and carbon dioxide move through that respiratory membrane. And before we go to all those details, I think it's important to realize that there is a system to the madness that seems to be happening in the respiratory system. Gases, oxygen and carbon dioxide, but air itself, through the process of ventilation, is always moving. We have to have a direction to these movements, so we cannot live with just haphazard movement of air or oxygen, carbon dioxide. So here is the key to this system. When we will talk about diffusion, I hope you remember way back from AMP1 that diffusion is representing a movement of, in our case, gases from a place of higher to a place of lower concentration. In our case here, we will translate it to a movement of place of higher to a lower pressure for oxygen and carbon dioxide. In the case of ventilation, air and all its component gases will naturally move to a place of high pressure to a place of low pressure. When we explain why oxygen is leaving hemoglobin in the tissues, we come back to the very same relationship, movement from a place of high to a place of lower pressure. And even when we explain the events inside a hyperbaric chamber towards the end of this lecture, this is the relationship, this is the driving force that will, I hope, bring a whole lot of clarity to all these events in the respiratory system. So let's take a look at ventilation first, the movement of gases through our conducting and respiratory zone. This is a summary of events during ventilation. So we do have inhalation and exhalation. And the question we're trying to answer, why is air moving into our respiratory system during inhalation? And how can we make air move out of our respiratory system during the process of exhalation? So looking back at that Boyle's law, for inhalation, we have to expand the volume of the thoracic cavity. And so through the use of skeletal muscles that we'll detail in a minute, we are going to lift up on the rib cage. We're going to flatten a diaphragm. And you see some of these muscles listed here. And we are expanding the volume of thoracic cavity. According to Boyle's law, this increase in the size of the thoracic cavity will lead to a drop in the pressure. The value read here is 758 millimeters of mercury. When you compare it to the air outside of our body, 760, here's that magic relationship. Air will naturally move to a place of high to a place of low pressure. And we have air streaming into our respiratory system. We have an inhalation just because we expanded the volume in the thoracic cavity and created a low pressure environment inside. Let's take a look at exhalation. A different set of muscles and events will lead to the decrease in the size of the thoracic cavity. This action will create a high pressure environment now inside our respiratory system. Here again, air will very naturally move from this place of high to the place of low pressure, and air is going to be streaming into our environment, and this is what we call exhalation. So pressure differences explain very naturally why air on its own moving in and out of our respiratory system. To create these movements, we need skeletal muscle to expand or decrease the volume of the thoracic cavity. So in the case of inhalation, let's talk about this first. It's kind of cryptic on this slide, but the external intercostal muscles will lift on the rib cage, and diaphragm will be contracting, so diaphragm will flatten, and that increases the volume of thoracic cavity for inhalation. To create an exhalation during quiet expiration, all we have to do is relax the external intercostals, relax the diaphragm, and through the action of no muscles, the thoracic cavity will slump back to its original volume, and this is what quiet exhalation, or in professional terms quiet expiration, is happening. So this quiet inhalation and quiet exhalation exchanges the air volume of roughly 500 milliliters, the volume of a regular water bottle. If we need more airflow into our respiratory system, some disorders, physical activity such as exercise will bring this about. We have labored breathing. Forced inhalation is created by a stronger contraction of the diaphragm, a stronger contraction of the external intercostals, and additional muscle force. Sternocleidomastoid scalenes, pectoralis minors, they will all be lifting the ribs, expanding the volume of the thoracic cavity even further. Therefore, more air will be streaming into our respiratory system during forced inhalation. When you take a deep breath, you're consciously start the action of these muscles, and you can invite a lot more air during that forced inhalation when you take a deep breath. With all that extra air in the respiratory system, we need additional muscle force to exhale all that air. So forced expiration follows forced inspiration. And here we do use muscles, abdominal muscles such as external oblique, internal oblique, rectus abdominis muscles will contribute to the decrease in the volume of the thoracic cavity. We do have internal intercostals as well to contribute to this. So these muscles will make the thoracic cavity volume decrease and help to create more pressure to eventually squeeze out, exhale out more air during labored breathing. This is the role of pleural membranes, which is very unique among all these thin membranes. So when you look at this cadaver transfer section here, you see the lungs and you see the rib cage, the wall of the thoracic cavity. Between these two large body parts, there's a set of thin membranes, the parietal pleura that lines the internal aspects of the thoracic cavity, and the visceral pleura that lines the surface of the lung, and in between them we have pleural fluid. When we expand the volume of the thoracic cavity, the pleural fluid develops a little vacuum and stretches the lungs, brings the lungs along with the expansion of the thoracic cavity, so our lungs are expanding as a consequence of that. Because the pleural fluid is an essential component in making inhalation happen through the expansion of the lung to follow the expansion of the thoracic cavity. If for some reason that pleural fluid is replaced with air, the disorder pneumothorax or collapsed lungs occur, and this is a best indication how much we need this pleural fluid. When we don't have it or it's replaced with air or blood, the lungs can no longer follow the expansion of the thoracic cavity. So no matter how much we're trying to inhale, the lungs will remain collapsed and no air will be flowing through the respiratory system on this side of the lungs. So the pleural membranes and the pleural fluid that they make are absolutely essential for inhalation. And you know, if we have no inhalation, we have no exhalation either, because there's nothing to push out from our lungs. And so the collapsed lung, pneumothorax conditions can be life-threatening. Patients may need to go on a ventilator to expand the lungs when we don't have this connection of pleural fluid between the lungs and the thoracic cavity. So we have looked at now all the gross and micro anatomical structures, not only within the respiratory system. We have listed a number of skeletal muscles that expand or shrivel the size of the thoracic cavity to bring about inhalation and exhalation. So our next look in the respiratory system explores why there's a natural flow of oxygen and carbon dioxide from airspace to blood, and from, in the case of tissues, from the cells into blood. Respiration gas exchange requires the movement of oxygen and carbon dioxide, and diffusion will be taking care of these. So let's review again that relationship, just how movement from place of high to a place of low pressure will bring about gas exchange. I skipped the previous slide for now because let's take a look at the numbers first here. Our topic is external respiration on this slide, so we are focusing on this portion. Here you see a cluster of alveoli and the blood coming to this alveolus. And so in the venous blood, we have an oxygen concentration of 40 millimeters of mercury. In the air that we inhale, we have an oxygen concentration of 160 millimeters of mercury. So first, where will oxygen go? Naturally, we don't need to put out directional signs, no need to text oxygen to move into the blood. In our alveoli, because of this concentration difference, oxygen will naturally move from a place of high to a place of low concentration. So we have oxygen streaming into blood vessels into the red blood cells. So the venous oxygen concentration can be measured. The previous slide was related to this 160 millimeters of mercury value. This can be calculated, and Dalton was the first person to realize that if you take air pressure 760 millimeters of mercury and take 21% of that number, which is 21% is the percentage of oxygen in the mixture we call air, 21% of 760 comes out to it as 160 millimeters of mercury. So this is the driving pressure for oxygen in the air that we inhale into the alveoli. So that's all that how Dalton's law relates to calculation of this number here. When we look at the movement of carbon dioxide, we're still in this external respiration portion of this slide, of this illustration. So in our venous blood coming to the alveolus, carbon dioxide concentration or pressure is 45 millimeters of mercury. In the air, we have only 0.3 millimeters of mercury carbon dioxide pressure. Yet again, this gas, carbon dioxide, will follow the driving forces of diffusion, moving from a place of high to a place of low concentration. So carbon dioxide very naturally will stream out of the venous blood here and stream into the airspace of the alveoli. And this is how we enrich the exhaled air in carbon dioxide. From the blood, we have gas movement into the airspace of the alveolus, and we accomplished gas exchange here. Oxygen zooming into the blood, carbon dioxide is zooming out of the blood because of these pressure differences. This illustration is now focusing on this lower part. This is the question how oxygen will move out from the blood to the tissues, first into the interstitial fluid, and how carbon dioxide moves from cells into the blood. This is the internal gas exchange or internal respiration. We have to have this movement of gases to deliver oxygen to the cells and to eliminate carbon dioxide from the cells. So let's take a look at the numbers. This is going to drive the movement. In the arterial end of the capillary, we have 100 millimeters of mercury oxygen pressure. The tissues, because cells used up the oxygen, the tissues have 40 millimeters of mercury oxygen pressure. So very naturally here too, oxygen will move from a place of high to a place of low concentration or pressure. So from a blood, oxygen will be diffusing into the interstitial fluid, and the interstitial fluid that's created by the bulk flow will have that high oxygen concentration delivering oxygen to the cells, where oxygen will move into the cells because of this pressure difference. How do we get rid of that carbon dioxide waste product? Our cells, because of constant production of carbon dioxide, create a fairly high pressure area, and so in the cells and in the interstitial fluid we build up carbon dioxide concentration. In the blood, the arterial blood, the carbon dioxide concentration is lower. So in this case too, carbon dioxide will stream from cells to interstitial fluid, into the arterial blood because of this pressure difference. So yet again, we have a movement from place of high to a place of low pressure. And so our cells keep getting rid of carbon dioxide because carbon dioxide will be moving into the blood, and blood will be taking carbon dioxide away to the place of alveoli where we already have seen carbon dioxide naturally will enter into the airspace. We have a little segment here on oxygen transport, and you are saying that, wait a second, in the blood chapter we already dissected hemoglobin left and right, and we looked at recycling there. So here our goal is yet again to ask the question why oxygen is moving off of hemoglobin in the tissues and why oxygen is moving onto hemoglobin into lungs. So to explain all this, we have this handy-dandy curve. So this is the best way to look at this curve. Think about oxygen pressure here as 100 millimeters of mercury. This is the pressure on hemoglobin inside arterial blood. When this blood comes to the tissues outside of arterial blood and outside of hemoglobin, we have 40 millimeters of mercury. In a very hard-working skeletal muscle, the oxygen pressure may even drop to 20 millimeters of mercury. So what is oxygen going to do on hemoglobin? Oxygen will see a difference, pressure difference from 100 millimeters of mercury in the blood to 40 millimeters of mercury or even 20 millimeters of mercury in the interstitial fluid and in tissues. And oxygen will wave goodbye to the blood because of this pressure difference. And yet again, very predictably, oxygen will move from a place of high concentration to a place of low concentrations, and oxygen will detach itself from iron, will move off of hemoglobin and diffuse into the tissues where there are much lower oxygen concentrations. So movement of oxygen even from hemoglobin to the neighboring fluids is explained by this pressure difference moving from a place of high to a place of low concentrations. We can force hemoglobin to release more oxygen. In addition to the pressure difference, the pH and the temperature has effect on oxygen movement. The higher the temperature, the more oxygen will be waving goodbye to hemoglobin and will be released for the tissues. Working muscles tend to, this is a really good example to explain this. Working muscles have higher temperature. Hemoglobin will give up its oxygen much more readily when blood arrives into a hard-working, warm skeletal muscle. Hard-working cells, skeletal muscles, also release acids. Lactic acid is a typical acid that accumulates in the tissue fluid, and that acidic condition also creates a favorable movement for oxygen from hemoglobin to the tissues. Buildup of carbon dioxide, and we'll see this just in a minute, creates carbonic acid. So lactic acid and carbonic acid from carbon dioxide contribute to this low pH. And low pH, again, encourages oxygen to move away from hemoglobin. And so oxygen released from hemoglobin can now go to those hardworking tissues. For the carbon dioxide transport, we have something totally different. We don't have a designated carbon dioxide transporter. Even this hemoglobin carries a small fraction, a minority of carbon dioxide. Most of the carbon dioxide will be transported in plasma in a form of bicarbonate ions. So this takes a chemical change. Carbon dioxide will be turned into carbonate ions, and we'll see just how this is done right away. So in tissues where carbon dioxide is made by the mitochondria, as soon as carbon dioxide enters blood, inside red blood cells there is this enzyme. So not only hemoglobin is found inside red blood cells, but this enzyme, carbonic anhydrase, and carbonic anhydrase will speed up these chemical reactions from carbon dioxide plus water, carbonic acid, and then bicarbonate ion will be created. And this is the transporter that will accumulate in the plasma. So this is our major carbon dioxide transporter. Thanks to the speedy action of carbonic anhydrase, this change occurs lightning fast. So carbon dioxide is changed into bicarbonate in the blood that is going through the tissues. When the same blood arrives into the lungs, carbonic anhydrase inside the red blood cells is going to reverse its action. Carbonic anhydrase in the blood that is streaming through the lungs will take bicarbonate and change it back into carbon dioxide. So inside lungs, we are exhaling carbon dioxide that was recreated from bicarbonate through carbonic anhydrase. And so this is an essential enzyme that allows a large amount of carbon dioxide to be transported in the plasma in the form of bicarbonate, and allows the return of carbon dioxide from bicarbonate when the blood is circulating in the lungs. So you would have to zoom in to see carbonic anhydrase action inside the red blood cell, whether in the lungs or in the tissues. And on the previous slide, a much larger view of these chemical equations are present. Here on this illustration, you can see the different direction of carbonic anhydrase producing bicarbonate when blood is flowing through tissues, and producing carbon dioxide when blood is flowing through the lungs. Here's the last gas law, Henry's law. Henry's law just states that the higher the pressure of a gas over a surface of a fluid, the more of that gas is going to stream into that fluid. And again, gas will move from a place of high to a place of low pressure. That generic explanation applies here as well. For hyperbaric oxygenation, we are taking advantage of this Henry's law. We can put patients or people into a tent that can be inflated to 3 to 4 times the 760 millimeters mercury pressure. This high oxygen pressure in the air inside this hyperbaric chamber will move even more oxygen into the blood. We are now raising the driving force. So oxygen here will move from a place of very high pressure to a place of low pressure inside the blood. And sometime when plasma oxygen carrying can be increased through this process, that a little bit of oxygen moving through a plasma can help people with carbon monoxide poisoning, some kind of a hypoxia during heart disorders, or even help to treat anaerobic bacterial infections because oxygen concentration is now increased in the blood as a consequence of raising the ambient air pressure and oxygen pressure in this hyperbaric chamber. The last part of our lecture here is about control of breathing. So we cannot control diffusion. We cannot control air pressures. The only thing we control in our respiratory system is the ventilation process, inhalation and exhalation. The control of inhalation and exhalation is possible because those movements are carried out by skeletal muscles, and changing the number of nerve impulses to those skeletal muscles can change their activity, can stop them, can make them contract stronger. So during normal quiet respiration and quiet ventilation, all we need is skeletal muscle action during inhalation. So in our medulla oblongata, this inspiratory area, this group of neurons will create nerve impulses 12-15 times a minute. So we can control this part of the medullary rhythmicity area, the pacemaker of our respiratory center. Even when we sleep, nerve impulses created 12-15 times a minute to make us inhale. And if you remember, quiet exhalation requires no muscle force, so the expiratory area neurons stay totally quiet. So the inspiratory area is giving the pacing to our inhalations. If you want to change that pacing, we have a pneumotaxic area in the pons that can extend the exhalation. The aneuploidy area can extend the inhalation length. Maybe the most important practical use of this is when we talk. The expiration or exhalation has to be extended, and the airflow continually need to be provided through our voice box. So pneumotaxic area is frequently disrupts our respiratory pattern, extending exhalation when we talk or sing. We can also have conscious influences on the inspiratory area. You can hold your breath, and so we can stop the pacing from this inspiratory area. We can consciously override the inspiratory area when we hold our breath, or at will you can speed up your respiratory pattern. You can start panting, which means 30 to 40 inhalations per minute. When you do that, again, you are overriding the inspiratory area. When you allow our bodies to take its natural rhythms, we are seeing 12 to 15 inhalations per minute, again, coming from this automatic pacing from the inspiratory area. If you remember, in our heart, we have the sinoatrial node providing pacing for our heart. The very same idea. A group of neurons create spontaneous bursts of nerve impulses that, in case of respiratory center, now go to the diaphragm and the external intercostal muscles to create automatic inhalation. The last comment I want to make on the chemical regulation of our respiration and our inhalation and exhalation. In the lab, we'll come back to this a little more. So, in addition to the effects of the nervous system, we do have ways to monitor carbon dioxide and oxygen content in our blood. It kind of makes sense that a buildup of this waste product, carbon dioxide, is a warning sign. It means that our respiratory center is not fulfilling its role. It's not working at high enough capacity. So what can we possibly do about that? So whether in our medulla oblongata, the chemoreceptors detect high carbon dioxide concentration, or in the peripheral chemoreceptors, such as the carotid bodies or in the aortic arch, if those chemoreceptors detect high carbon dioxide concentrations, these neurons will create a nerve impulse, and that nerve impulse will reprogram the medullary rhythmicity area. So we have sensory neurons detecting changes in carbon dioxide levels, and the buildup of carbon dioxide is the most significant signal. In terms of nerve impulses, from the peripheral chemoreceptors, from the aortic arch, it's cranial nerve 10 that will take the nerve impulses to the medulla oblongata. From the carotid bodies, it's cranial nerve 9 that takes nerve impulses to the medulla oblongata. And so this is how our medullary rhythmicity center can be reprogrammed. The pace of inhalations will increase, and we can also move the body, switch the body into forced exhalation and inhalation, into labored breathing, if carbon dioxide buildup is detected either in the medulla oblongata or in these peripheral areas where we monitor blood carbon dioxide concentration. So this is the end to our respiratory system. I hope everything is fairly clear. As always, don't ever hesitate to ask questions. In our case, please message from within the class shell, and I'll respond as soon as I can. I'll see you guys in a little bit.